Okay, so today's session is going to be about stem cells, and we're going to have a look at what stem cells are, how they work, the benefits of them, and how we can use them in medicine and treating people with different diseases, genetic or non-genetic diseases, and treatments in those realms, and then some ethical issues on the use of stem cells. So stem cells are a type of cell that's basically unspecialized, and a stem cell can become different types of cells. So what we're going to look at first is a totipotent stem cell. And so what you'll sort of hear me using actually during this session is a few different terms. You'll hear things like totipotent and pluripotent, and then unipotent. And all this means is the prefixes, so like toti, pluri, and uni, tell us what types of cells these stem cells can differentiate into. So basically, stem cells are undifferentiated cells which can differentiate into specialized cells. So they become specialized cells. And each one of these three different categories of stem cells, they can become either every type of cell possible in the human body, or less, or even really more specific. So, for example, a unipotent stem cell, and you also will see multipotent. Unipotent generally becomes one type of specialized cell, whereas multipotent can become a few, and toti and pluri are the ones that can become the most. So that's what they mean, and we'll talk about that in a bit more detail, but I thought I'd cover that quickly now. So yeah, totipotent stem cells. Totipotent stem cells, they're able to mature, but able to, when we say differentiate, it basically means that different genes are switched on and off and they become a different type of cell. They're able to mature into any type of body cell. So a multicellular organism, like a human, for example, are made up from lots of different types of cells. And these different types of cells are specialized for their function. So we're made up of a liver, which has got liver cells in, muscle cells, white blood cells, red blood cells, heart cells, you name it. We've got lots and lots of different types of cells. And all these specialized cells originated from stem cells. So when an egg gets fertilized and we get an embryo, those first early cells, when we get to about day 4 or 5 in an embryo, are called stem cells. And these stem cells have that potential to become every single different type of cell inside the human body. And it works the same for all mammals. So stem cells are unspecialized cells, and they can develop into other types of cells. So the stem cell in that 4 or 5 day old embryo could become a liver cell, or a muscle cell, or a white blood cell, any type of cell in the human body. And what stem cells do is they divide to become new cells. And upon dividing, they become specialized. So these totipotent stem cells, these stem cells that we have in the early embryo that can become any type of cell in the human body, they're not around forever. They're only around for a few early days of the embryo, and then those cells start to become specialized straight away. And some might stay stem cells, but they might become multipotent stem cells, and they start to specialize, and that's how it works. So on dividing to become new cells, the stem cells become specialized. All multicellular organisms have some form of a stem cell. So plants have some form of stem cell, humans do, all mammals, reptiles, everything, all multicellular organisms have some form of a stem cell. And the stem cells we're talking about right now, these stem cells are found in the embryo. And when they become all the specialized cells needed to form a fetus, so that early embryo is made up of all these stem cells. And we also find some stem cells in some adult tissue. So you as a human adult now, you do have some stem cells still inside you. And these adult tissues, where we've got some stem cells, they will become specialized cells that need to be replaced. So, for example, we have stem cells in the intestines, which constantly replace intestinal epithelial cells. And basically, because the way the epithelial cells regenerate and get used so quickly, it doesn't work via the process sort of mitosis that we would normally use. So stem cells actually replace those cells, similar to red blood cells. Because they don't have a nucleus, they can't go through mitosis. So red blood cells are replaced by stem cells all the time. So totipotent stem cells are only present in mammals in the first few cell divisions of an embryo. So these are only present in those first few cell divisions of an embryo. So totipotent is the most potent, can become every single type of cell possible in the body, and that's only those first few cell divisions of an embryo. So up to day 4 or 5 of an embryo, we still have these totipotent stem cells. So that's really, really important. So it's those first few cell divisions of an embryo. After this point, the embryonic stem cells become pluripotent. They can still specialize into any type of cell in the body, but they lose the ability to become the cells that make up the placenta. So your totipotent basically have that ability to be any body cell plus a placenta, cells that we need for a placenta. And then after those first few cell divisions of an embryo, they become pluripotent, and that is any body cell. Again, so any body cell, but this time not the placenta. It cannot, these cells cannot form a placenta. And obviously that's because by this point, the beginnings of the placenta has already started to grow, and the placenta then is going to carry on growing via itself. It doesn't need stem cells because that basis of the placenta is starting to grow. And then the embryo doesn't need to hold these stem cells that can make a placenta because the placenta is already off growing by itself. So stem cells present in adult mammals are one of two. They are either multipotent or unipotent. So you have totipotent cells in the very, very early first few cell divisions of an embryo. Then we have pluripotent, and that will help us form the fetus, and we still have some as the fetus is growing. And then as the fetus gets more developed, there's less and less pluripotent stem cells. And then as a fully grown adult mammal, we don't have pluripotent stem cells. We have the option of two others, and that is multipotent stem cells and unipotent stem cells. Multipotent stem cells are able to differentiate, again, become specialized. They go from being a stem cell to a specialized cell into a few different types of cells. So one example would be bone marrow. Bone marrow is a source of stem cells, and from that we can make all the different types of blood cells that we need. So for example, red blood cells and white blood cells, and it's also a source of platelets. But yeah, bone marrow is an example, so it can make both red and white blood cells. And obviously we have multiple different types of white blood cells as well. So our bone marrow can make all of those. Or you might find in your body unipotent stem cells. These can only differentiate into one type of cell. So for example, there is a unipotent stem cell that can only divide to produce epidermal skin cells, and that makes up the outer layer of your skin. And basically we have to have stem cells to replace the outer layer of our skin, because our outer layer of our skin basically gets broken down so quickly, and because it's exposed to the environment, the way it gets broken down, it doesn't work by replication going through mitosis. So actually stem cells, we have a source of stem cells that basically continually are replacing that epidermal skin cell, and they get replaced that way rather than actually via mitosis. And that's an example of a unipotent stem cell. So toti is absolutely everything, every single possible cell plus a placenta. Pluri is every single cell in the adult body apart from placenta. Multipotent is a few different types of cells, and unipotent, one type of cell. So stem cells differentiate to become a specialized cell. And they do this because different genes are expressed. So we've spoken about gene expression, genes being switched on, genes being switched off. And stem cells become specialized because during their development, they only transcribe and translate part of their DNA. So they obviously hold all the DNA, all DNA possible to become every single type of cell inside the human body. And then they only transcribe and translate part of that DNA. So stem cells contain, like I just said, the same genes. All stem cells have exactly the same genes as each other. But during development, so during embryonic development, we're talking about here, not all of them are transcribed and translated. So not all of those genes are transcribed and translated. So they're not all expressed. Tip to remember, transcription is when our DNA is copied into mRNA, and translation is when proteins are produced using the code in the mRNA. So under those right conditions, some genes are expressed and others are switched off. So we have to have the right conditions provided. And some genes are expressed, whilst others are switched off. And this is at the beginning of that differentiation process. So if about a stem cell to become, for example, a liver cell, certain cells, certain genes are going to be expressed, the ones that help it become a liver cell, and ones that would maybe cause it to become a heart cell or any other type of cell are going to be switched off. mRNA is then only going to be transcribed from those specific genes which are now expressed. The mRNA from these genes is then translated into proteins. So genes are switched on and off. mRNA is only transcribed from the specific switched on expressed genes. That mRNA from those specific genes is then translated into proteins. These proteins then that are produced are able to modify the cell. They determine the cell structure and they are able to control the cell processes. So this also includes the expression of more genes, which produces more proteins. And changes to the cell produced by these proteins cause the cell to become specialized. And these changes are really, really, really difficult to reverse. So once a cell has specialized, it stays specialized. Later on, we'll talk about actually how we, in a lab, and we've discovered how we can actually force some cells to go backwards and actually re-become stem cells and become pluripotent. But naturally in your body, it's really difficult to reverse these changes. So our cells, once they have specialized, they stay specialized. So an example would be a red blood cell. So red blood cells are produced from a type of stem cell that we find in the bone marrow. Red blood cells have lots of hemoglobin inside them, and they have no nucleus. And remember that no nucleus is to help make room for the red blood cell to carry more hemoglobin. And what happens is the stem cell produces a new cell in which the genes for hemoglobin production are expressed. Other genes, such as those involved in removing the nucleus, are also expressed, and many other genes are not expressed, so we switch them off. And this results in a specialized red blood cell. And that happens every single time. So those stem cells in the bone marrow are going to express the genes for hemoglobin production and express the genes for removal of the nucleus. Switch off the other genes, and then we end up with the red blood cell being produced. So cardiomyocytes, this is an example that we're going to look at specifically. Cardiomyocytes are heart muscle cells, and we can make these from unipotent stem cells. So these are produced from unipotent stem cells. So red blood cells are an example of ones that are made from multipotent stem cells. So our bone marrow is an example of a source of multipotent stem cells, whereas cardiomyocytes can be produced from unipotent stem cells. So as I just said, cardiomyocytes are heart muscle cells, and they make up a lot of the tissue in our hearts, make up that cardiac tissue. In mature mammals, it was originally thought that they can't divide at all to replicate themselves. So we think they can't divide. It's thought that they can't divide to replicate themselves. That's where we are. So we can't regenerate heart muscle. So for a long amount of time, because we think that these heart cells can't divide to replicate themselves, going through processes like mitosis, for example, we thought that we are not, so because of that, we thought that we weren't able to regenerate our own heart cells at all. So the process and the thought was that if our heart became damaged, so for example, by having a heart attack, all the cells become worn out through age, those areas of damage or areas of worn out heart muscle would not get replaced. However, recent research has suggested that our hearts do have some regenerative capability. So where we're at with this now is some scientists now think that old or damaged cardiomyocytes can be replaced by new cardiomyocytes, and this is derived from a small supply of unipotent stem cells in the heart. So we've now thought that actually there's a small amount of unipotent stem cells in the heart that can replace these cardiomyocytes. The problem is with this is that some researchers think this process could be constantly occurring, something that it might not be constantly occurring. So we know that there's some unipotent stem cells there, but we're not quite sure how fast this replacing of damaged or old tissue is happening. So some scientists believe that it's really, really slow process, and it's actually possible that some cardiomyocytes are never replaced for a person's entire lifetime, and that some of those heart cells have been there since they've been born. Other scientists think that actually it's occurring a lot more quickly, and so that every cardiomyocyte in the heart is replaced several times in a lifetime. We don't know. I mean, they're two literally polar opposite views, and we do not know yet. So again, this is an area that would have more research put into it. So we can use stem cells to treat human disorders. As we know, stem cells can divide into a range of specialized cell types. So what doctors and scientists have thought is, well, we could use these to replace cells that have been damaged by illness or injury. Because if a cell has already been damaged by illness or injury, it can't go through, it can't divide and replicate to make a new one because it's been damaged. So you'd have to wait for other ones to make up the area, and sometimes it just doesn't happen. So you can have an area where we don't have cells or tissues being replaced. So the process and the idea was, well, could we use stem cells? Some stem cell therapies already exist. So it's not this sort of really sci-fi into the future, or this might eventually happen one day. Some do already exist. And they're mainly for diseases that affect the blood and the immune system. So bone marrow, which we've spoken about quite a bit already, contains stem cells, and these can become specialized to form any type of blood cell, which is really, really good because we can use bone marrow as a transplant. So we can have bone marrow transplants, and these can be used to replace faulty bone marrow in patients that produce abnormal blood cells. So the stem cells in the transplanted bone marrow then divide and specialize and produce healthy blood cells in the recipient. This technique's been used really successfully to treat leukemia, which is a cancer of the blood or bone marrow, and lymphoma, which is a cancer of the lymphatic system. It's also been used to treat some genetic disorders, such as sickle cell anemia and severe combined immunodeficiency, also known as SCID. Bone marrow transplants, you have to be a bone marrow donor. You can be living. It's not a donation that has to come from someone who's deceased. So you can be a living bone marrow donor. You have to be quite young to be it. I think you need to be under 35 or 30 to be a bone marrow donor. And you have to be very healthy, and you have to also be a match. So you have to be a tissue match, a bone marrow match. And it's not necessarily has to be someone in your family. Sometimes you will match with people in your family, sometimes not. And basically what happens is a large needle, it's very painful, but it's not too risky, goes in through normally the groin or the top of the hip, through the bone and into the bone marrow, and then the bone marrow is taken from the healthy person and then given to the ill person. Healthy person has a longer recovery because obviously now they've got a reduction of bone marrow, which means that their blood, so like the white blood cells, the red blood cells, aren't going to be replaced as quickly. And that bone marrow will get replaced because they're healthy, but this is why someone has to be really, really super healthy to be a bone marrow donor, because if you're not, it would be a very risky procedure to have done, because you're basically depleting your stem cell supplies for then making new blood cells. And you do make new bone marrow, but obviously that will take time. But the person receiving it actually gets better very, very quickly. So SCID, we'll just have a quick look at what SCID is. And SCID is severe combined immunodeficiency. SCID is a genetic disorder and it affects the whole immune system. And what it is people with SCID have a really poor functioning immune system. And that's because their white blood cells, which are made in the bone marrow from stem cells, are defective. And what this means by them being defective, it means they can't defend the body against infections by identifying and destroying microorganisms. So SCID sufferers are extremely susceptible to infections. So they can get poorly very, very quickly with something just as simple as a common cold. So we can treat people who have SCID. And when we say people, it's normally children and babies. Because these people often would not live very long without the bone marrow transplant, because they would die very early from picking up something as simple as a common cold. You have to wait until the baby's old enough to be able to manage a bone marrow transplant, but then you have to keep them healthy enough in that time, waiting until they're old enough and strong enough to receive a bone marrow transplant. So the treatment with bone marrow transplant replaces the faulty bone marrow with the donor bone marrow, and the donor bone marrow contains stem cells without the faulty genes. So the issue is in their stem cells in their bone marrow that would have the gene would be faulty. So every single new type of blood cell, that white blood cell they make, would be defective. Whereas in the new bone marrow, it doesn't have the faulty genes that cause SCID. These would then differentiate to produce functional white blood cells, and these cells can identify and destroy invading pathogens, so then their immune system functions properly. SCID, with it being genetic, when we've talked about pre-genetic implantation diagnostics, so people who have IVF and test the embryos for genetic issues. If SCID runs in the family, you can test for SCID on embryos to make sure that the embryo you're implanting doesn't have SCID. Some people do this and then have something called a savior baby. So they might have a baby who's got SCID, and they need to find someone with a bone marrow match to then donate bone marrow to their baby who's got SCID. You're most likely to find a match with siblings, because siblings have closer tissue type matching than either parent. So if either parent aren't a match, then they often use siblings. And so if there's not a living sibling, some parents choose to then go on and have another child to then use their bone marrow to save their other child, which is called savior siblings. And this is very controversial and a massive ethical issue. And it has to get past lots of ethical boards. So some places won't do the post-implantation genetic diagnostics because of it being for SCID, for making a savior baby. So it is very complicated, but it's something you're interested in. It's worth going and having a bit of research and having a look into. But it is very complicated, and it goes on sort of a case-by-case basis. Some countries allow it more than others. But there is a lot of ethical issues, and obviously ethical issues further down the line. And that baby that's born might not actually be a bone marrow match. The baby who's got SCID might die in the process while you're waiting for this other baby to be big enough and healthy enough to donate the bone marrow. then the long-term psychological effects this has on both children, it's very complicated. That's sort of just a bit of an aside. So we can use, stem cells could be used to treat other diseases. So currently scientists are researching the use of stem cells as treatments for lots of conditions. Ones I'm going to go through are ones that they're researching at the moment. They're not available yet, but some are undergoing clinical trials. So some people will be being treated with these things at the moment, but because it's a clinical trial, it's not readily available treatment. They've just taken part in a clinical trial to try and see if the stem cells work. So spinal cord injuries, stem cells could be used to replace damaged nerve tissue. These could also be used in spinal cord cancers as well. Heart disease and heart attack damage on hearts, so stem cells could be used to replace damaged heart tissue. Bladder conditions, stem cells could be used to grow whole bladders, which are then implanted in patients to replace diseased ones. And in respiratory diseases, donated windpipes can be stripped down to their simple collagen structure, and then we can cover them with tissues generated by stem cells, and this could then be transplanted into patients. The idea of that is we could reduce the risk of rejection if we use the patient's own stem cells to do this. Also, windpipes are quite hard to donate, they're very complicated organs. And then finally, organ transplants. Organs could be grown from stem cells to provide new organs to people on donor waiting lists. But again, these are all ones that are being researched at the moment and some are going through clinical trials but aren't readily available. So there is a huge benefit to using stem cells in medicine. So people who make decisions about the use of stem cells to treat human disorders have to consider the potential benefits of stem cell therapies. So they can save many lives. So like I was saying about the organ transplants, people waiting for organ transplants often die before a donor organ becomes available. And stem cells could be used to grow organs for those people who are waiting for transplants. They can also improve the quality of life for lots of people. So stem cells could be used to replace damaged cells in the eyes of people who are blind, for example. So we're going to look at the 3 types of stem cells that we can get from adult tissues or from embryos. So to use stem cells in research or in medicine, scientists have to get them from somewhere. So there's 3 main potential sources of human stem cells. The first source is adult stem cells. And these are the stem cells that you can get from the body tissue of an adult. So for example, adult stem cells are found in bone marrow. They can be obtained in a relatively simple operation, and they have very little risk, but they do involve quite a lot of discomfort, like I was talking to you earlier about the bone marrow transplants. Issue with adult stem cells is they're not as flexible as embryonic stem cells. They can only specialize into a limited range of cells, not all body cell types. They're multipotent, and actually some are unipotent. So they don't have that flexibility. We can only use them for a small range of things. The second source of human stem cells is embryonic stem cells. We obtain embryonic stem cells from embryos that are at an early stage of development. These embryos have been created in a lab using in vitro fertilization, and the egg cells are fertilized by sperm outside of the womb. Once the embryos are approximately 4 to 5 days old, the stem cells are removed from them, and then the rest of the embryo is destroyed. And the rest of the embryo has to be destroyed because when we remove all those stem cells, we're basically removing the ability for that embryo to then divide and develop into a fetus. It can't. So by removing those stem cells, we're destroying the embryo. Embryonic stem cells can divide an unlimited number of times and develop into all types of body cells because they're pluripotent. And our third source is induced pluripotent stem cells, iPS cells. These are really new, and there's a lot of research going into those at the moment. So iPS cells are created by scientists in a lab. And what it is the process involves us reprogramming specialized adult body cells into pluripotent stem cells. So, for example, taking skin cells and turning them back into pluripotent stem cells. That's the idea. So, how this works is the adult cells, so for example, the skin cells, are made to express a series of transcription factors that are normally associated with pluripotent stem cells. And there's a lot of different ways these can be done. I'll give you an example of one in a moment, but there's lots of different ways. But you have to stress them out a lot. You have to put a lot of stress on them for them to take up these transcription factors. The transcription factors cause the adult body cells to express genes that are associated with pluripotency, and these genes would have originally have been switched off. And it's very hard to swap that, like we said earlier. So one of the ways that these transcription factors can be introduced to the adult cells is by infecting them with a specially modified virus. And the virus has the genes coding for the transcription factors within its DNA. And when the virus infects the adult cell, these genes are passed into the adult cell's DNA, meaning that the cell is then able to produce the transcription factors. Other ways involves sort of stressing them out with heat or electric shock. That's just one that you can have a bit of an idea about. You don't really need to know that in too much detail. Induced pluripotent stem cells could become really useful in medicine, and they're really good in research at the moment. At the moment, the main issue with them is that we need more research into how similar they actually are to those true pluripotent embryonic stem cells. And we need that before we can properly use them. And there's also the risks of them. Do we know how they work properly? Are they as efficient? Do they work as well? Because ethically, lots of people would say, yes, this is a lot better than using embryonic stem cells. But in the long run, are they as good? Do they last? Is using them when we've got these viruses involved going to open up chances of people getting more ill? Can we get them to the right part of the body? Do they later, because we've messed about with their genes, are they more likely to become carcinogenic and actually develop into tumours and cancers? So again, we have to take that into consideration. Finally, a few ethical issues that we'll look at surrounding the uses of embryonic stem cells. So obtaining stem cells from embryos created by IVF raises ethical issues because the procedure results in that destruction of an embryo, and that embryo could become a fetus if it was placed in a womb. So some people believe that at the moment of fertilization, an individual is formed that has the right to life. So they believe it's wrong to destroy the embryo because we're destroying a life or a potential life, depending on how people view this. Some people have fewer objections to stem cells being obtained from egg cells that haven't been fertilized by sperm, but they've been artificially activated to start dividing. This is because the cells couldn't survive past a few days and wouldn't produce a fetus if we placed it in a womb. Other people think that scientists should only use adult stem cells because their production doesn't destroy an embryo, but adult stem cells can't develop into all the specialized cell types that embryonic stem cells can. And so this is where induced pluripotent stem cells could be really good because they have the potential to be as flexible as embryonic stem cells, but as they're obtained from adult tissue, they don't have the same ethical issues around them as embryonic stem cells do. It's also possible that the iPS cells could be made from the patient's own cells. So these iPS cells, which would be genetically identical to the patient's cells, could then be used to grow some new tissue or an organ that the patient's body wouldn't reject, because rejection of transplants occurs quite often and it caused by the patient's immune system recognizing the tissue as foreign and attacking it, and people have to be on immunosuppressant drugs for a lot of their life. The decision have to be taken very strongly. We have to take lots of people's views into place. In the exam, you could be asked to evaluate the use of stem cells in treating human disorders. So you sort of need to know all the pros and cons around the use of stem cells and the benefits of one type of stem cell to the benefit to compared to another, and then the ethical issues. So an example of an exam question would be, scientists are currently exploring the potential for the use of stem cells in medicine. A, it gets you to explain one way in which stem cell therapy is currently being used. I'm not going to go through every single possible answer you could give, but I'm going to go through one example. And I'm going to use the bone marrow examples. So for your first mark, you could say stem cell therapies are currently being used. So they're currently being used for some diseases affecting the blood and immune system, because that will cover your bone marrow, affecting the blood and immune system. That'll get you 1 mark because we've said one place that it's being used. Now I'm going to say what we're going to use. What is this source of stem cells? So bone marrow contains stem cells that can differentiate into any type of blood cell. You get another mark for that. So explain one way in which stem cell therapy is currently being used. Well, it's currently being used for some diseases affecting the blood and immune system, first mark. Now we're talking about the sources of the stem cells, so that gets you a second mark. The bone marrow contains stem cells that can differentiate into any type of blood cell. Okay, so how are we using it? So we can have bone marrow transplants. And they can be used to replace, I'm going to use leukemia for my example, replace faulty bone marrow in patients with leukemia. Which is a type of cancer of the blood or bone marrow. That will get you 1 mark. So we've said where it's being used, diseases of blood and immune system, we're using bone marrow. How are we using it? Well, bone marrow transplants could be used to replace a faulty bone marrow in patients with leukemia. How does that help them? That will be the last 4th mark. So the stem cells in the transplanted bone marrow divide and specialize to produce healthy blood cells. That will get you the 4th mark. So the second part of this question, explain why some people object to the use of embryonic stem cells in treating human disorders. You start off with this by saying how we get the embryonic stem cells. So obtain embryonic stem cells involves the destruction of an embryo. First mark. Second mark, why? Why do some people object to this? So we said it involves the destruction of an embryo. Some people believe that embryos have a right to life. Oops. And it's wrong to destroy them. And then the final part of the question, it may be possible to use induced pluripotent stem cells, iPS cells, instead of embryonic stem cells to treat human disorders. Describe how induced pluripotent stem cells can be produced. So 4 marks for this. First mark, we want to mention that reprogramming. So I'm going to do this one as a bullet point. So we're going to say iPS cells are produced by reprogramming specialized adult body cells to become pluripotent. That would be your first mark for this question. Your second mark, how? How does this work? So to do this, the adult body cells are made to express a series of transcription factors. So the adult body cells are made to express a series of transcription factors. I'm going to put TF, transcription factors that are associated with pluripotent stem cells. Third mark, the genes code for the transcription factors are introduced to the adult cell's DNA. So here we go. So we've got genes code for the transcription factors are introduced into the adult cell's DNA. And this happens using a modified virus. That has the genes in its DNA. And now finally, there's just going to be one more exam question I want to go through before we finish this session. So finally, I just want to go through this exam question, which is looking at SCID. So SCID is a severe inherited disease, and people who are affected have no immunity. Doctors carried out a trial using gene therapy to treat children with SCID. The doctors who carried out the trial obtained stem cells from each child's umbilical cord. Give 2 characteristic features of stem cells. They are undifferentiated, or you can say they can differentiate, develop into other cells, totipotent, multipotent, pluripotent, anything around that. I'm just going to say that. No, undifferentiated. And the other part is that they will continually keep dividing, replicate, or replace themselves. So the So the doctors mixed the stem cells with viruses. The viruses had been genetically modified to contain alleles of a gene producing full immunity. The doctors then injected this mixture into the child's bone marrow. The viruses that the doctors used had RNA as their genetic material. When these viruses infect cells, they pass their RNA and two viral enzymes into the host cells. One of the viral enzymes makes a DNA copy of the virus RNA. Name this enzyme. And that enzyme, as you should remember, is reverse transcriptase. That'll get you 1 mark. The other one would have gotten you 2 marks. So the other viral enzyme is called integrase. Integrase inserts a DNA copy anywhere in the DNA of the host cell. It may even insert the DNA copy in one of the host cell's genes. The insertion of the DNA copy in one of the host cell's genes may cause the cell to make a non-functional protein. Asks you to explain how. So this is going back to that idea of how some proteins might not be functional, gene mutations. And what could happen is it could alter the bases, causing a frame shift. A frame shift, so alters bases causing a frame shift, and a frame shift causes a different sequence of amino acids to be present in the polypeptide. Which can affect the tertiary structure. That would give you 2 marks. Some of the children in the trial developed cancer. How might the insertion of the DNA have caused cancer? So you're just getting how might this happen? So have a think about what might have happened. So something to do with tumor suppressor genes. So we could have had an effect on the tumor suppressor gene. So it could have inactivated a tumor suppressor gene. Which would lead to an increase in the rate of cell division. We'll get you 2 marks. And then finally, this is the last part of this question. 5 out of the 20 children in the trial developed cancer. Although the cancer was treated successfully, the doctors decided to stop the trial in its early stages. They then reviewed the situation and decided to continue. Do you agree with their decision to continue? And explain your answer. Now you can say yes or no, that doesn't matter, but you need to give a good reason why. And you don't get a mark for yes or no, it's your marks for stating yes or no. So I'm going to go through both. So if you said yes, you need to justify why. So yes, because SCID patients are unlikely to survive anyway. They have a poor quality of life unless we treat them. So they're unlikely to survive without treatment. As we saw there, the cancer was treated. The cancer was treatable. And only affected 25%. If you said no, here is why you would have to say no. So the risk of developing cancer is high. at 25%. And the cancer may reoccur. And not be successfully treated. So that is everything about stem cells. And that goes into topic 8, 8A of AQA A-level biology under mutations and gene expression.